Good afternoon and welcome everyone. So before we start the session, I want to acknowledge the country. So on behalf of TRI, I pay our respects to the Turrbal and Jagra peoples, the traditional custodians of the land and waters of the Brisbane area. We recognize their connection to land, water, and culture. We pay respect to elders past, present, and emerging. We acknowledge the stories, traditions, and living cultures of Aboriginal and Torres Strait Islander peoples and commit to fostering a culture of learning from and working with First Nations peoples in the spirit of reconciliation and access to justice. So hello everyone, my name is Kavita Bisht and I'm an early career researcher at Marta Research. It is my pleasure to have Professor Flavia Huygens and Paul Carboon today for our first research translational seminar. So we will hear from these speakers first, and then we'll have 10 minutes at the end for Q&A. So please keep yourself muted and turn off your video camera. And at the end, you can either type your questions on Zoom chat functions, or you can unmute yourself to ask a question. So our first speaker today is Professor Flavia Huygens, who is Executive Director, Co-founder, and Chief Scientific Officer at Microbio. Flavia's 30-year teaching, research, and commercialization career has been focused on molecular microbiology with a particular focus on human pathogens. Flavia brought together emerging technology in molecular microbiology, genomics, proteomics, and bioinformatics to invent InfectID, a diagnostic test that identifies bacterial, fungal, and viral pathogens. Flavia is co-inventor or inventor of a growing number of patents, 4 of which are related to InfectID, which is currently being commercialized by Microbio. Then our second speaker is Paul Carboon. Paul is a leader in biotechnology innovation. He has 25 years of commercialization experience in the technology sector and has a deep understanding of commercial management, and product development. Paul has successfully taken 4 greenfield projects to market with products that continue to achieve multi-million-dollar annual revenues. He currently serves on OSMed Biotech Advisory Group and is a founding advisor with the Rich Track program. So please welcome Professor Flavia.
Thank you very much for that very comprehensive introduction. And I am very excited to be talking to you today because it is the first of the series, and very excited to bring Microbio's technology to the attention of the audience. So first and foremost, as the title suggests, our talk today is really about translating research into a commercial product. And you might be asking yourselves, well, how does that actually work in the real world? Oh, sorry. So the fundamental thing to bear in mind here is that 2 heads are definitely better than 1. Now, there's a bit of a joke between Paul and myself as to which is the better head or the bigger head, but let's leave that out for you all to decide. So as per the introduction, I have been based in research and teaching at QUT for the last 20 years, but have been working in the field of in vitro diagnostics and molecular microbiology, particularly relating to infectious diseases, for 30 plus years. And the concept that underpins InfectID was actually started, or the fruition of that concept was started more than almost 20 years ago now as well. So it has been a while. This has been a long journey. Just to bear that in mind. And I guess from developing the product, which was the initial concept of the foundation of what would become InfectID, which was then obviously followed by you need to prove the technology, you need to prove the principle and prove it and prove it and prove it. And so that took a number of years of working, I guess, to get to the point of actually doing pilot studies. So needed to have collaborators in the field of infectious disease diagnostics. And in this particular instance, that was Pathology Queensland. And so they have been walking along this journey with me from the very early days. Then, of course, following that, once we had proven the technology in some pilot studies, we had to further do a lot of research and development, or R&D, followed by design and development. So this terminology was all new to me, being a research and teaching academic at QUT. A whole new language had to be learned and a whole new understanding of this world that I had entered. And of course, regulatory submission, that is a beast all on its own. And I will not spend too much time elaborating on just all the intricacies of acquiring regulatory approval for products, particularly in the in vitro diagnostic space. So at the end of that sort of first from concept through to R&D, D&D, and regulatory approval, we obviously side by side had to thinking about, well, how is this going to actually fit inside of a company? And so in the next slide, I will talk to you about, starting back in 2016, where I met Paul for the first time. And he sort of really laid the foundation for really making me think about, how do I take this concept that has been proven in those early phases with collaborators such as Pathology Queensland, and how does that then move on to the next phase in getting a company established? So that is really where our paths crossed for the very first time. And as per the introduction with Paul's background in the space, that was really such a crucial step for me personally to meet Paul, but also to take that first view or glimpse of, well, now what is the next step and how do we continue taking this product forward? So that's when Microbio was established. And of course, the next challenge was, well, where is the intellectual property sitting for this technology? And I won't spend a lot of time on that. So we have got time at the end for any questions. So please make sure that, feel free to ask any questions about any of the points that I raised here and what Paul will raise as well. So that interface between developing the product, building the company, and then going into commercializing the product was really the whole journey of starting in the early days to where we are currently now, and then also looking to next steps. So after securing the IP, obviously we were able to start recruiting staff into the company, which then launched our R&D and our pilot study and our R&D phase and onto D&D as well. And so we are now at the point where we are really seriously looking at commercializing the product. And so again, this is where Paul's expertise and know-how and guidance really comes into play with regards to, well, this all costs money. So how do we go about securing funds to do all of this work, to make sure that the technology is carried forward with its development, and also with regards to how do we actually take it through the regulatory approval, et cetera, et cetera. So a little bit about the evolution of Microbio, as I stated already. I met Paul back in 2016. And that is where we crossed paths because I had done some early proof of concept for what would become InfectID, and was ready to apply for an industry partnership program grant, in particularly with our defence, so the DMTC program, but I needed an industry partner. And literally went onto Google, literally went onto Google and typed in Australia IVD partners and companies, because being a defence project, they required the industry partner to be onshore. And literally 2 names popped up. And so the first name was literally the one that I cold called, and Paul answered the call. And so that is literally how things happened. I know it sounds very weird and bizarre. But that was Paul then came up to Brisbane, which we met, and we started down the journey of discussing what the technology is, what its potential is, Unfortunately, at that point in time, time ran out for the grant application, so that didn't go forward. But in 2017 and 2018, actually, we reapplied and it was successful. So again, the common theme here being just persevere. Okay, and I'm going to keep saying that. So that was pre-foundation. Foundation, obviously, in 2017 was when the IP was secured and the company was founded. So securing the IP and putting that into the company was really the foundation of Microbio. Subsequently, from the early days, pre-company establishment and also post-company establishment, as mentioned already, we've had subsequent patents that are in various phases across the world with regards to their examination and granting phases. Back in 2019 and onto 2020 and 21 was really our full steam ahead R&D and D&D phases, in particular for two of our products, which I'll mention in the next slide. And where we are right now as well, Paul will elaborate on that very shortly with regards to our QMS certification and the next steps towards our commercialisation of our products. So if we only focus on the last 2 years, that being 2020 and 2021, what did we achieve as a company? So InfectID is our brand name for our products, but of course we're looking at a whole spectrum of infectious agents. So our flagship assay being InfectID BSI or bloodstream infection, and sepsis being a medical microbiologist by trade, knowing that sepsis is deadly and a huge challenge when it comes to diagnostics and rapid diagnostics and testing turnaround time. That has really been my passion over all this time, is there has to be a better way to do this in order to save lives. So InfectID BSI R&D was completed in 2020-21. And during that time, we also completed our full, what we call our preclinical, our pilot study. So that was with our collaborators at the Royal Brisbane Women's Hospital Emergency Department and Mackay-based Hospital Emergency Departments as well. But I mentioned DMTC in the early stages of me meeting Paul and the reason why our paths crossed. So we were successful in obtaining a collaborative grant through DMTC, and that was for our InfectID SSBA product. So the R&D for that study and that grant was completed in 2021 in collaboration with DMTC, but also Defence Science Technology Group as well. Of course, COVID came along, and so we pivoted towards developing and applying our technology to COVID in the early months of 2020. And again, through our partners and our collaborators, we did some preclinical studies on our COVID test. They are called InfectID COVID R&D. Again, can answer questions on those later on. Subsequently, in last year, we gained European approval and regulatory approval for our COVID tests, not only in the EU, but also in India. We've also set up a manufacturing line for some of our core ingredients for the assay and have very recently been awarded an ANGC grant that will enable us to initiate this work inside of TRI. And that is exactly why we've moved to TRI as well, because this was the first step along our journey for manufacturing. I mentioned QMS. We obtained our stage 1 approval in December last year, and we are working very furiously towards our stage 2. And together with that, we've also developed the InfectID client software. So my last bit before I hand over to Paul is just what are we currently doing this year? So I mentioned the QMS. So this is our ISO 13485 certification, which we will be audited on in early April. And we're also currently investigating scale-up manufacturing, and not only in Australia, but also internationally. And we are very proud that we will be presenting 4 abstracts on our core flagship assay, which is our InfectID BSI, in the upcoming European Congress for Clinical Microbiology and Infectious Diseases in Lisbon in April this year. We are also working very hard at the same time on getting our InfectID BSI ready for regulatory approval in Europe and India as well in May of 2022. And I mentioned the AMGC grant that we have obtained. So we are moving towards setting up manufacturing inside of TRI. And we also expect to submit our submission or our application for regulatory approval for InfectID BSI to the TGA towards the end of this year, beginning of next year. And of course, we're keeping our eye on the FDA as well and working on this process. I will now hand over to Paul, who will be talking to you about what our goals are for the next couple of years. So over to you, Paul.
Excellent. Thank you very much, Flavia, and great presentation. So I guess just touching on what Flavia talked about from the beginning, having a pure commercial brain is never going to get you a product to market, and having a pure scientific brain is never going to get you a product to market. You have to marry the two of those and really work together and trust each other in what you're doing. So it's been an amazing journey with Microbio thus far, and I think we've developed one of the best teams I've ever been part of, and very exciting. So from our point of view, we've key strategy has been to raise as little money as we need to get us through to the next milestone. In that way, you then continue to keep value in the company for the existing shareholders and obviously the founders. So, you know, one of the key strategies is to always, where you can, to gain grant money from either federal or local or whatever, wherever you can get it, really. And one of the things you'll need to continue doing, and being academics, most of you, I'm sure you're well aware of applying for grant money, but yeah, really part of the process is to work with the grant bodies to continually develop and pitch for grants. It's actually a very, very good exercise, especially with the accelerated commercialization and those type of grants. They really help you build out your commercialization planning into the future. And, you know, for people who don't have a great deal of commercialization experience, I think working with those types of groups is really, really important to do. But yeah, so working through the whole process, it continues to develop a plan around how you're going to commercialize your technology. And the plan always is to raise more capital. Well, okay, raising capital, as I said, is it's time consuming, it's hard, and often, you know, what you think you've created value is not necessarily what the investors are looking for from that value point of view. So our plans really are to, yes, continue down the capital raise pathway, and what most commonly would be called a Series B capital raise. Series B capital raise is generally, you know, the minimum would be 5 million up to 20 million, and that'll secure the company for at least 5 years or 3 to 5 years. So that's sort of a potential opportunity for us next year. What we have to do this year, though, is we have to increase our value, and that's really what those previous couple of slides that Flavia showed you, was trying to get our value up to, you know, 50, 70, $100 million. Therefore, raising 20 doesn't dilute everyone out of the picture and end up with nothing. So more attractive, though, is a cornerstone investment from an industry partner. So what does that mean? That means we take our technology, which we've already got registered or approved on certain countries, and partner with someone who's already got existing distribution networks and sales networks and whatever else, and you know, work with them to get your product into the market. And that really can be a very rapid process. So one of the key things you always want to be looking for is who are those partners and who can we partner with to get that type of transaction completed, which is beneficial to us and beneficial to them in the short term, generates us revenue, reduces the dilution component of it, but also then allows us to secure the company and do further product development. So, you know, and Australia has lots of fantastic support around that. So the other one is an exclusive technology license. Now, this is the big boys. This is where you get bought out, or you get that major milestone. I can tell you they are a lot further down the road than you ever think they might be. thinking a multinational is going to come in and do a license deal with you within the first 5 years of being in the market, I think that's a bit ambitious. So, you really plan for that, but it's always something that's out there a little bit further, and either one or two have to occur before, and secure the company and get into market and generate revenue and have all of those clinical stories and have that, that big market pull happening before one of those multinational life science companies will come in. They'll come and buy you beforehand, but you won't get any value out of it. So, and then there's obviously the ASX or stock market listing. Now, that is always an option, and I think it's a very attractive option. We saw, I think around about 17 listings in the last year and a half, quite successful listings in this space on the ASX. So it's always a potential. We have some issues with that. Is that one of the things being an ASX listed company, you have to tell the market what you're doing all the time. So that's not really something you want to do in a product development sense. So might just is that all I was going to talk about, Flavia? Is there another slide?
Yep, I think, Paul, if you can continue with our takeaway messages.
Sure. So no, we're running out of time, but key takeaway messages is you need to. Going back to 2016 when Flavia and I first met, we talked about what is the maximum cost price of a manufactured product for our sepsis product. We talked about why would people want to buy the product? What are the key important reasons that people would buy our product? And they became our mantra, right? Cheap, quick, broad pathogen range, high specificity, high sensitivity, open platforms. I don't know how many times we've said those words, Flavia, but I think, a million times a day sometimes, isn't it, right? And drilling that into your staff and drilling it to everyone that you talk to about what are the key things that are going to get your product into the market, and really focusing on those, but understanding those up front and validating them with the market is really important. Managing intellectual property, I think there's a significant caveat here that I really want to point out to you. And I talk to lots of startups, and they go, we've got to go and file a patent. And I go, why? And they're like, well, we need to protect your IP. But do you actually know what your IP is? And a lot of cases, you have to really get into the product development stage, understand what your core IP is, and then file a patent, or not, right? Sometimes you want to hold back from the market and from others, context that is really your know-how, and a lot of that's walking around in Flavia's brain at the moment. But you get the context. You might not always want to file a patent. So rushing into patent filing is one thing, but really making sure you understand what your pathway is. And as pointed out in the slide earlier, we've filed, I think, three more patents since the original patent was published. And just incredible that we can get to that point of being able to validate that as part of our product development journey. Securing funding, wow, that is one of the hardest things to do in the world. I mean that absolutely. And you really have to plan what your key objectives are. You meet someone today, 2 years' time, they might want to turn around and invest in you. And if you said, well, you're going to do these things, they'll remember that. And you really need to understand when and how and why you're going to be securing funding, and what the outcomes of that are for the investor, and when they're going to get their return, and all of these things. It's quite difficult to do. Develop, verify, and validate your product. As Flavia mentioned, R&D, D&D, conformance assessment, all of these QMS, all of these terms that many researchers do not understand. You need to get across that. You need to make sure you can get the right people to help you do that. Gaining regulatory approval. This is not for the lighthearted. It really isn't. You have to partner with someone who really knows what they're doing in that space. Just trying to negotiate with the FDA or, the European Union or whoever directly yourself when you've never done that before, you will fail, in my opinion. Relationships with key industry partners, I can't stress that enough. And, the whole point of, getting a product to market is you need to be able to find people that can manufacture it at those cost prices, and as I said, partnering with other people to take your product to market into the international world is probably the best way to do it. So, I'll stop there.
Yeah, thank you, Paul. And so just finishing off with the very last slide there, and I think if you haven't heard it already, it's about identifying where the gaps are and seeing where you can find people to fill those gaps. So knowing your capability and your knowledge base, and then knowing where the gaps are and finding people. So whether that be staff, and, we are very proud of our current staff. It has been a journey to get all these amazing people together, but we're not done yet, of course. And having consultants that are really value add. Sometimes you sort of have pitfalls along the way, and we've experienced those. But again, we are very, very fortunate to have amazing consultants that are working with us, advisors, collaborators, etc. And of course, this all takes time. It takes time, it takes effort, it takes patience, and of course, perseverance. So never, ever give up. And you do have to kiss a whole lot of frogs along the way. Thank you so much for your attention, and would welcome any questions that you might have.
You can ask questions via chat or by Zoom. So I'll start asking you one. So now with the COVID, we know PCR, rapid antigen test. So how different is InfectID? Like, what's the difference?
Yep, sure. Thank you, Benny. Very good question. So as you saw on the slide, we've got two COVID products, InfectID R and D. So no, it doesn't stand for research and development. So InfectID R, it actually looks at the SARS-CoV-2 virus when it is actively replicating. And so we are looking at parts of the subgenome that is only present when the virus is internalized inside of the host cell and replicating. So people would think, so what? What does that mean? Well, any other PCR test that's out in the market at the moment only tells you, yes, there is genomic message of the virus. It doesn't tell you whether that virus is replicating, making copies, in other words, inferring whether the patient or the person could be shedding live virus particles. So that is our unique test. The D test is also very unique in the sense that it is universal. It picks up all variants. And because we are targeting our core technology, which is the most conserved part, yes, the virus does have a conserved part of its genome. And so every 2 weeks, we are cross-checking, and Alex sitting in the audience is our guru at doing this cross-checking. He runs through thousands and thousands of SARS-CoV-2 genomes every 2 weeks to make sure that our DNR tests are still valid. So I hope that answers your question.
That's it. Thank you. Do we have any questions from the audience for Paul or Flavia?
I do have one. I'm probably going to ask a very naive question.
No such thing.
I'm very naive on the field of commercialization anyway. Okay. So in the beginning of your talk, you mentioned, Flavia, that you have to kind of like see where the IP stands. Mm-hmm. What do you mean by that for someone who has no idea what you're talking about?
Can you repeat the question?
Yes. So the question there for the audience on Zoom land was about understanding what is meant by where the IP stands, as I mentioned very early on in this presentation. So for us, it meant, because it was the foundations of this IP was whilst I was an academic inside of a university or an academic institution. Mm-hmm. And so I had spent many, many years working with the commercialization arm within the university to really progress the product and the technology, etc. Unfortunately, that just took, X amount of time, and it was very challenging. Again, as Paul mentioned, getting funding, building those relationships with industry, a hugely challenging process. So the IP was always embedded inside of the university. So if that's the case for you, it's starting that conversation either with the commercialization arm of your institute or your organisation, and seeing whether they are able to assist you in progressing that product through all of these phases of research, development, et cetera, proof of concept, pilot studies, all of the above, engaging with industry, blah, 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 blah. But essentially, don't give up if that doesn't happen is the key message here, because again, through that building of relationship with your commercialization arm or commercialization office, in your institute, you could then see if they can't take it all the way through, that you can request that IP to be either assigned to yourself in your own personal capacity. So that is essentially what I did after many years, was to obtain the foundation background IP assigned to myself, and then that was put into the foundation, which became Microbio. Yeah, but of course that's just an example. It doesn't have to be like that every single time. Okay. Yeah.
Just in addition to that, Flavia, one of our future product sets, we've examined over 280,000 patents to make sure that we've got white space. So you can't pretend that I'm unique and it's all going to be. You have to do the hard yards. You have to do the work. It's just as simple as that.
Yeah, exactly.
Thank you.
Good question. No, good question.